All right, so this is station 2, mitosis notes. So make sure that on the correct page in your notebook, you write this purpose. Explain the importance of mitosis and the steps of the cycle, meaning from interphase to cytokinesis. So why is it important that cells divide? Well, you have to replace damaged or dead cells. This goes through mitosis. So if you look at this picture, every time that you get an ouchie hurty on your skin, you're going to need new cells to replace it. Things like your esophagus and your intestinal lining actually replaces itself every couple of days. So your cells are constantly dividing. You're also going to need it to grow. So this is a fertilized egg. So this was way back in the day, you were a single cell big, and now you are a bazillion cells. This is also because of mitosis. So as you can see, the function of mitosis is twofold: to replace and to grow. Finally, meiosis, which we'll talk about a little bit later, is to produce reproductive cells, your baby-making cells. If you're a girl, you produce eggs. If you're a boy, you produce sperm. This is through a slightly different process. It has some similarities to mitosis, but I'm going to tell you, you're probably going to mix them up. So start coming up with some ways to remember. I think of it, mitosis deals with your toes, your body cells. Meiosis deals with your baby-making cells. So here's a comparison of mitosis and meiosis. Again, we're going to cover this guy a little bit later, but mitosis creates body cells, as in like your toes, mitosis. A fancy word for body cells is somatic cells. So this is a summary slide. I'm going to go through each of the parts. You're going to start with 46 chromosomes, okay, and I'll tell you what chromosomes are. And you're going to start with what we call diploid, di meaning two, just like disaccharide. Then it's going to divide into two daughter cells, which are also going to be diploid. All of these cells are going to be identical. So you're going to have end result of two identical diploid body cells. Meiosis creates sex cells. The fancy pants word for that is gametes. You're going to start with the diploid number, if you're humans, 46, and you're going to end up with 4 haploids, so twice as many cells as mitosis. And you're going to end up with 4 genetically unique haploid sex cells. So if this is occurring in a boy, these are sperms. If this is occurring in a girl, it's eggs. And if it's a plant, pollen. Fun fact, pollen is plant sperm. Now you know. So this image is showing you kind of the life cycle of a cell. You start with something called interphase, which has 3 sub categories, and then you go in through cell division. M-phase meaning mitosis, and then this thing called cytokinesis. And we're going to break down what occurs in this interphase. Draw this in your notes. I'm going to be explaining it. Pause the video, draw that bad boy. So in interphase, most of the time is spent in this phase. The cell is growing, it's developing, it's getting its final form, and it is preparing for division. So G1 is the first phase of interphase, and that is growth. You can think of G standing for growth. S stands for synthesis. Synthesis means to make. I'm going to need to make new DNA for when I have my baby cells. The DNA is going to be copied. Then during G2, right here and right here, the cell prepares for division. So you're like double checking chromosomes, making needed repairs, you're making more organelles. The cell is also going to be growing just a smidge. So sometimes the cell is going to be entering this thing called G0 from G1. That's what it's showing right here. And they're just not going to divide. They're just going to be there. So think about your body. And think about what cells don't really regenerate themselves. If you damage it, you lose it forever. Think about it. It's your nervous system. So people with brain damage or spinal cord injuries, they're paralyzed for life. There's some evidence that suggests that maybe they just divide really slowly, but for our purposes, as a rule, things like your nervous system, those cells hang out in G0, those neurons that we talked about last semester. So when the cell divides, it must be copied in that S phase. This fancy pants word for copied is replication. So you're going to have to make a new copy of DNA for your new cells that you produce, period. We'll revisit this in meiosis, but you're going to need to make more DNA as you make more cells. So now we're going to talk about what happens to the DNA. So the first thing that's going to happen is DNA opens up. As we talked about before, the DNA is kind of like a twisted ladder where you have hydrogen bonds just kind of sticking these two guys together, so they're going to split apart. Step 2, you're going to have matching nitrogen base pairs. So the A is going to bond with another T that's floating around. The C is going to bond with a G. Think about this. Where did these guys come from? Nucleic acids. They came from your food. Remember, everything that we eat that is living has nucleic acids. That's why we don't care about that when we did our diet essay. You're going to be getting more A's, T's, G's, and C's from the food that you eat. So step 3, the identical DNA molecules become made because these guys are going to stick together. So you're going to open it up like a zipper, and then it's kind of like the size of your zipper, bond with another zipper to make 2 different molecules, one old and one new. So sketch this picture by interphase, just so that way you know what the cell looks like. So you have a nucleus, and you have something called chromatin. So chromatin, remember, is like really messy DNA. It's like just relaxing, it's chilling. Eventually it'll turn into that chromosome X form. Then we also have centrioles, which I'll talk about, which are right here. And keep in mind, other organelles are in there, mitochondria, endoplasmic reticulum, but we don't care about them, so this model simplifies it a little bit. So pause the video, draw it in your notes. So mitosis, remember, is division of body cells, somatic cells. It's, by definition, the division of the nucleus. The nucleus is what we care about. There's 4 phases: prophase, metaphase, anaphase, and telophase. We're going to go through each one by one and talk about what happens. A mnemonic for this is PMAT. So prophase. The chromatin condenses to become chromosomes. So remember, this looks like messy yarn. It's going to become those like X forms, really neat and organized. The nuclear membrane is going to be disappearing. And the centrioles, which are an organelle, are going to move to opposite sides of the cell. These centrioles make something called spindle fibers. Think of it like fishing line. And those things are made out of microtubules, which are a protein. So that DNA can take on a couple different forms. Chromatin is uncondensed. So remember, to be condensed means to put closely together. Think about condensed soup. And it just looks really messy. I draw it as scribbles. So the DNA is loose like an unraveled extension cord, or if you've ever seen Christmas Vacation, when he takes out all of those knotted up twinkle lights. The chromosome is a condensed form. It's nice and organized. And it's like an extension cord that's all wound up. So here's a cell. Here it's all messy, kind of hard to see. And now they turn into those characteristic X forms. So draw this in your notes so that way you remember chromatin versus chromosomes. There's a lot of C words in here, and you're probably going to screw these up. So let's figure that out now. Okay, now draw this under prophase. So again, we have the nuclear membrane disappearing. That's why I drew it as a dotted line. I have my chromosomes here. I have 3 of them. Notice how I drew one big, one medium, one small, so I can differentiate. I have centrioles right here, and I have these little hairs coming from them. Those are the spindle fibers. So during metaphase, the chromosomes line up along the center of the cell. Metaphase, think middle. The spindle fibers attach to another C word, centromere, to move them. The centromere is at the very center of that cell. So looking at this picture, here I have them all lined up, and these spindle fibers are attached to the very center of those chromosomes. So draw this in your notes. Draw me a cell. Draw me the centrioles on either side. Spindle fibers attach to the center of those chromosomes at what's called a centromere. So the halves of the chromosome are called sister chromatids. Okay, these bad boys are identical. Pause the video, draw this in your notes. Make sure that you have something in your notes to clarify where the centromere is. It's at the very center of the X. Anaphase. The sister chromatids separate at the centromere. So here's another great picture. Chromatid, chromatid make up a chromosome, the left and right halves, and they're attached at that centromere. The chromatid is half of a chromosome, and in mitosis, they are identical. They're twin sisters. So during anaphase, those chromatids are going to be slowly pulled to opposite sides of the cell, as you can see here. These go left, these go right, or up or down, or however it is that you're drawing your cell. So draw this in your notes. Draw me a cell, draw me 2 centrioles, and draw me chromosomes. Actually, no. Okay, here's a technical thing. Once the chromatids are not separated anymore, they're called a chromosome, just draw me half of an X. Okay, half of an X being pulled to the other side. And make sure you have this in your notes. Differentiating between a chromatid and a chromosome, labeling the centromere. Telophase is the last phase of mitosis. The spindle fibers are going to go away. They're going to break apart. They're going to get chopped up. Remember that they're proteins, so they're going to be put back into their monomer form. The chromosomes are going to return to chromatin. So remember that messy, tangled up yarn looking stuff, because it's kind of relaxing. It doesn't need to move, so it can just take up all the space it needs. The nuclear membrane is going to reform to protect that DNA. And so that's what it's showing here. It kind of looks like a butt or a Mr. Peanut. You are breaking apart those cells. So draw this in your notes. Draw me like a peanut shape or an 8 shape. Draw me the nuclear membrane reappearing. That's why I drew it dotted. Don't show me spindle fibers because they're going away. And show me the 3 chromosomes in each of the cells. So in cytokinesis, that is the last phase. That is division of the cytoplasm. It's technically not considered mitosis. So animal cells are going to pinch to create something called a cleavage furrow. Cleavage just means crack, friends. Plant cells are going to develop something called a cell plate. These are kind of going to be dividing from the outside in and these from the inside out. So draw this in your notes. The chromatin is going to return to chromosomes will turn back into chromatin, and here I have 2 identical looking cells. So remember, back when we started, we also had 3 chromosomes. They looked a little bit different because they were replicated chromosomes, because I went through that S phase. This bad boy actually goes back into G1, and once it goes through S phase, it will have the other half of its X. So I have identical daughter cells that are also identical to the parent cell. So let's recap. Mitosis is division of the body cells, like my toes. Your toes undergo mitosis. You undergo interphase, meaning G1, S, and G2. Then you're going to go through PMAT, prophase, where the nuclear membrane starts to disappear, metaphase, where the chromosomes line up in the middle, anaphase, where those chromosomes start to be ripped apart, telophase, where I begin to have 2 new nuclei form, and cytokinesis, where they eventually split. Then I might have some G0 going on, where the cells are just going to hang out like the neurons in your body. So now I want you to go through these notes and write your 3 house questions: a Google, a Doodle, and a Noodle. Let me know if you have any questions. Okay, bye.